And during my PhD, I became really fascinated by the RNA that is found in extracellular vesicles, and especially the mechanisms that make these RNAs end up in these tiny bubbles. Because these pathways appear to be highly complex and to me also highly fascinating. So some early papers have discovered a couple of ways RNAs can be routed to extracellular vesicles. For instance, by the action of proteins that specifically bind to these RNAs and then recruit them to multivesicular bodies or to the plasma membrane where vesicles are being formed. But there also appears to be a lot of passive loading of RNA protein complexes, but also of free RNAs into extracellular vesicles. And I think we're only scratching the surface of all the pathways that are out there. And I think this paper is a good example of that. So I was immediately struck by the title because of my interest in RNA sorting. But here they study autophagy, which is a way for cells to overcome starvation by recycling nutrients. And they do this by degrading defective proteins and organelles in a structure called the autophagosome that then fuses with the lysosome to degrade cellular material. And actually, if you think about it, both autophagy and EV release are ways for cells to degrade and recycle cellular material. So what they also have in common is that both autophagy and EV formation rely on the rearrangement of membranes, as you can see in this schematic here. You have the formation of these phagophores that form into autophagosome, and this is just as EV formation regulated by a vast number of proteins with regulatory functions. And one of them, a key protein in this paper is called LC3, which is modified by many different proteins, ends up on the phagophore and is cleaved into a shorter isoform called LC3-2. And indeed, autophagy is classically known as a process of degradation of cellular components, as you can see in this cartoon here. But it's also becoming apparent that autophagy is also an unconventional secretion mechanism, at least for a couple of proteins that have been uncovered till now, such as cathepsin, IL-1 beta, lysozymes, and a couple of others. So the authors took this as a starting point to explore what other proteins are also released via this pathway and that we don't know about. And the way they approach this is by what I think is a very elegant method. So they try to characterize the autophagy-dependent secretome by making use of a fusion protein of LC3, which is tagged with a biotin ligase from bacteria. So during autophagy, this LC3 fusion protein ends up in the autophagosome. Proteins are then biotinylated and are then released via autophagy-dependent secretion. So the secreted proteins are specifically tagged with something you can detect. And what I forgot to mention last time is that in many of these assays, they use bafilomycin and chloroquine to prevent the degradation of these materials in the autolysosome and instead routing them into the secretion pathways. So they show here by a Western blot with an anti-streptavidin that both in cell lysates and in culture medium, there are a vast number of proteins that are tagged by this method. And the proteins that are tagged, they differ from a tagging with a soluble biotin ligase, which you can see in the middle of the plots. They also show that many LC3 interacting proteins are tagged via this method. So this is a streptavidin affinity purification, where they show that the P62 and optin, I don't know this protein actually, in ATG7 are specifically tagged by this method in cells. So the system seems to be working. So then they go on with a quantitative proteomics approach in which they isotope label the soluble biotin ligase and the LC3 coupled biotin ligase with light and heavy isotopes, and then they look at differential incorporation or differential tagging of these proteins. So they detect over 350 different proteins, of which 31 proteins are enriched in the LC3 biotin datasets, which you can see in the heat map on the right. And besides autophagy-related proteins, they surprisingly also find EV-enriched proteins and also RNA-binding proteins. In figure 2, they take it one step further and show that these proteins are really released into EVs. So they isolate EVs from these cells by pelleting at 10,000 G, then 100,000 G, and all the soluble material is precipitated by TCA. And here on this plot on the left, they show that most of the LC3 biotin labeled proteins are detected in the 100K pellets. And these include the classical EV markers, but also LC32 itself, which is quite surprising. They also show that LC32 co-fractionates with EV markers in a sucrose density gradient. So really showing that it is associated with these floating extracellular vesicles. Another nice assay that they employ is a protease protection assay. They treat the EV pellets with trypsin in the presence or absence of triton, in which they show that LC32 is resistant to degradation in the absence of triton. So it's really enclosed on the inside of extracellular vesicles. And then in figure 2G, they perform a transmission electron microscopy to show that LC3 is really localized in the multivesicular bodies of these cells. They do a little bit more work on these multivesicular bodies in figure 3 using a fluorescence microscopy approach. So they transfect a construct called mCherry Rep5 mutant, which impairs endosomal trafficking. So this leads to enlarged endosomes with intraluminal budding, and this makes it probably easier to detect LC3 in these endosomes by fluorescence microscopy. So they transfect this construct into different lines of 293 T cells. One is wild type, the other one is ATG7 deficient. So this is a protein that is required for LC3 lipidation, and ATG14 is also present in the autophagy pathway, but in the degradative autophagy. So here are the images, and they show that both in the wild type cells, but also in the ATG14 knockout cells, there is a co-localization of LC3 with multivesicular bodies. But this co-localization is absent in the ATG7 knockout cells, which indicates that LC3 lipidation is really required for recruiting it to multivesicular bodies. They also show that this is the case in wild-type cells, which I will skip now in sake of time. So in figure 4, they connect the dots between these findings and earlier figures. So they hypothesize that LC3 conjugation may be required for protein packaging into extracellular vesicles. So they do more quantitative proteomics on extracellular vesicles from wild-type cells versus ATG7 knockout cells or ATG12 knockout cells. And in the schematic on the left, you can see that both ATG7 and ATG12 are present in these autophagy pathways. They're both in both pathways. And what they find is that there are various proteins enriched in EVs from wild-type cells relative to the different knockout cells. And there's actually quite a lot of overlap between the different knockouts and also with the earlier approach they took in figure 1. So many of the same proteins pop up. And by doing Go analysis, they show that many of these proteins have RNA binding properties and also even DNA binding properties. So they hypothesize that LC3 may be required to capture these proteins at the limiting membrane of the multivesicular body and incorporate them into extracellular vesicles. So then figure 5, this is where we left off last time because of the trolls, but I'm happy I can continue. So in figure 5, they show that both SAF-FB and RNPK, two proteins found in the proteomics approach with RNA binding capacity, co-fractionate with extracellular vesicles, as you can see on this Western blot. And by doing different pull downs, they really show there is an association of these proteins with LC3. And also that these proteins are not degraded during starvation, as you may expect. That may happen during autophagy, but no, they're not degraded. They're really released via extracellular vesicles. And figure 5F I found was really convincing. So they showed that the incorporation of of SAF-B and RNPK is really dependent on ATG7 and ATG12. So on the left side, you see in these knockout cells, both SAF-B and exRNPK are expressed like in wild-type cells, but they don't end up in EVs, as you can see on the right. And in figure G, they do a quantification of this, and they really show that if you have deficiency for ATG7 or ATG12, that there's no LC3 or SAF-B or RNPK that ends up in extracellular vesicles. So then they name this process LC3 dependent EV loading and secretion, or LDELS, which is not very catchy, but I think it really pinpoints what it means. Then in figure 6, they address what the effect of ATG7 and ATG12 is on RNA incorporation into extracellular vesicles. So they perform RNA sequencing on wild type and both of the knockout cells, and they show that these different knockouts, they don't really greatly affect the biotype distribution in cells, but it really has a dramatic effect in extracellular vesicles. For instance, there's seems to be an increase in tRNA and a decrease in snow RNAs. And actually, snow RNAs are the most affected RNA biotype in these knockout cells. We show here in a pie chart and also in the heat map on the right. And for the people who are not that much into RNA, so snow RNAs are non-coding RNAs that play an important role in the nucleus. So they're part of protein complexes that modify ribosomal RNAs, which allows them to properly fold. And it's kind of surprising that you find these nuclear RNAs in extracellular vesicles, but this is not the first instance in which they found snow RNAs in EVs. So there somehow seems to be a mechanism that enables this to happen. But then again, yeah, this is where it sort of stops for the RNA part of the figure. In figure 7, they go a little bit deeper into the mechanism behind these shuttling pathways, and they hypothesize that pathways that may be involved in multivesicular membrane curvature, which is of course required for EV budding. So do these pathways also play a role in LDLs? And is there a role for NSMase2, which you all know converts these lipids in multivesicular bodies to make budding occur? So they start by treating the cells with a compound named GW4896, which is a well-known inhibitor of NSMase2 and partially reduces EV release. And indeed, this reduces the amount of LC3 and also general EV release. So there seems to be a link there. And in figure C, they take a complementary approach by inhibiting these NSMases by short hairpin RNAs. And they sort of show the same effect, which they quantify in figure 7D. So if you have ATG7 knockout or an NSMase knockout, you have a massive reduction of LC3 release and also of SAFB and RMPK. Somehow they didn't quantify ALEX and CD9, which they do show in the plot in figure C. But I think this clearly shows there is something going on with the NSMases. And then another part that I didn't completely understand, so via a bioinformatics approach that they didn't really specify, they find this neutral sphingomyelinase activation associated factor called FAN. So that is an LC3 associated regulator of NSMase2. And again, they show by short hairpin knockouts of this gene that indeed FAN has leads to a reduction of SAFB incorporation and also the incorporation of RMPK and LC3, which also quantified in figure G. But what is really convincing, I think, is that if they mutate the LC3 interaction domain, which is found on the FAN protein, they can also really block the release of SAFB and RMPK. So it's really the interaction between FAN and LC3 and probably the downstream effect of NSMase2 that blocks the release of these different proteins. And again, on the right side, this is quantified. So this is end of the results, and the first time I read it, it sort of puzzled me because you have all these proteins that are interacting together, and I took the opportunity to summarize this into a model. So I think what I got from the paper is you have LC3, which through the action of ATG7 and ATG12 becomes lipidated and ends up of the limiting membrane of the multivesicular body. And here it sort of docks CEF-B, which can bind SNO RNAs, small nuclear RNAs, and also RNPK, which has a broader range of non-coding RNAs it can bind. And it also acts as a docking station for FAM, which can activate or maybe recruit, that was not really clear to me, NSMase2, which then helps in the membrane curvatures. So this whole bunch of proteins is then ending up into an exosome or another kind of vesicle, and is then released into the extracellular space. I hope this sort of concretely summarizes the paper. And then to finalize, I had a couple of unanswered questions, which may help to start the discussion later on. So I was wondering, is there basal LDLs activity in every cell, like there is basal autophagy activity, or does this only come into play when autophagy is increased, such as in cancer cells? I'm not sure if we should address these one by one, or if I should go through some of these questions and then have a plenary discussion about it.
Yeah, I'm happy to do it one by one if that seems appropriate.
Yeah, I think that'd be best. And I just want to say that anybody who wants to speak up, let me know. We can unmute people, so it's not a problem.
So while we initiated this study kind of under serum starvation conditions, purely from the perspective that we felt that we wanted to induce the autophagy pathway to study secretory autophagy, but also because it kind of alleviates some of the issues associated with proteomics of the secretome. And we didn't at the outset know that we were necessarily going to get proteins in extracellular vesicles. We kind of assumed that we were going to get soluble proteins like had previously been characterized. So we did this under serum starvation conditions. That said, we do observe this under what I would call basal conditions, full serum conditions. And we find this process of LDLs is modestly suppressed by rapamycin treatment. I think we would like to go back and look at taurin and maybe some more potent inhibitors of mTOR and activators of autophagy to study this a little bit more in depth. But certainly we do note secretion under full serum conditions.
Okay, yeah, that's really clear. And yeah, you already talked about some other inhibitors. So I was also wondering if you don't block the autophagosome lysosome fusion like you do in most of the experiments here, would you still be able to detect LDLs or?
So we have some of these studies ongoing in the lab right now. We're kind of wrapping up a paper kind of relating to this. But I'll speak a little bit about some of the candidates in this paper. We found that indeed some of these RNA binding proteins are released under treatment with baflomycin and chloroquine. But I believe that these drugs will have very significant impact on the entire process itself. I think there's a lot of work that needs to be done in this area, trying to dissect how things that interfere with endosome and endolysosomal pH or acidification really impact the secretome and exosome secretion or extracellular vesicle secretion itself. So yes, these things are secreted under conditions of lysosomal inhibition.
All right, that's a clear answer. And then I had a third discussion point. So do you think, oh, sorry?
Sorry to interrupt. I think my my mentor wants to say a point.
Oh, yes, of course.
All right, working on it. Let's see.
Okay.
If somebody can get to
I made
it
it
before me. There you go.
Okay. I just wanted to clarify, and Drew can actually interject if I'm wrong, but I don't think for any of the proteomic studies or any of the studies of the secretion, we did not block the lysosome with baphlomycin or chloroquine. They were done in steady state. But as Drew pointed out, that these things are actually, it doesn't matter whether you block or you don't block, they seem to be secreted. What is interesting, and I think I think one of our feature is that when you do block the lysosome, there's an entirely new kind of proteome that we're essentially trying to get a grip on. But we did serum starve for most of this, most of the studies, but there was no lysosomal block. We wanted to keep it, keep the pathway intact as much as we could.
Okay, then I probably misread that. Sorry.
Okay.
And then third point that I wanted to bring up. So, do you think there could be a signaling function for these non-coding RNAs that are recruited by this pathway, or is this more to maintain homeostasis in the cells? What do you think about that?
I think at least our impression of this process is, why would you if it didn't have a signaling rule, why would you secrete it and not degrade it? And so in our minds, that that argument suggests that probably this is having some sort of role, but we're not completely sure at this point. That said, so we're currently investigating these things, and some of the non-coding RNAs implicated in our pathway have been shown to be transferred and the like. But this is definitely an active area.
Okay. Yeah, that were all my points for now, but maybe we can open the discussion floor.
Yes, maybe I can join with a question. I've also put it in the chat box, but I was wondering, do you have any evidence that the secretion is first by a fusion of an MVB with an autophagosome and then fusion of this amphisome with the plasma membrane, or it's directly via the MVB?
Yeah, I can answer that unless Jay wants to. Our genetics our deletion of, or exosome secretion is essentially unaffected by autophagy knockouts that block the formation of the autophagosome, including ETG14 and FIP200. And thus, because of this, you can, it strongly suggests that fusion of an autophagosome with the endosome forming an amphisome is not required for this process, and thus we do not believe that it's the secretion of these factors is coming from an amphisome.
Okay, yeah, great, thanks.
So, Erez, did you want to ask your question or make a comment? I think you had a comment here. maybe not. So I also had a question that came from somebody whose microphone is not working. So the question here is that this person asks, do you have, and it's Yvonne actually, she asked, it looks like there's a non-coding RNA preference here. Is that real? Is that really what's happening? Is it mostly non-coding RNAs? And if so, why? Or is this some kind of a function of the library selection?
This is, I mean, it's a question we've admittedly racked our brains with. Is this something specific to our pathway, or is there some sort of association with how we've prepped the library? And I think that it's probably a little bit of both, but there has been a number of studies showing that snow RNAs are secreted and packaged into exosomes. I can think of Matt Shirtliff's paper in PNAS from Randy Sheckman's lab, who we talk with from time to time. But I think it could also be colored or regulated by the fact that this serum starvation process may actually be kind of specifying things a little bit in our system as well, because serum starvation is a potent inhibitor of growth pathways and the such, and snow RNAs being involved in ribosome biogenesis and a modification to specify the translation and expression of certain proteins. So there might be a selection for those things to be removed from the cell under conditions in which maybe the cell doesn't want to express quite as much material, translate as much protein. So that's kind of how we view it. I think that we would really like to go back and broaden our search of RNAs that are released, because as you know, in this process, you kind of look for what you are hoping to find. And so we are currently trying to look at some of the other RNA species that may be regulated inside cells, as well as outside in the secretory pathway. Does that answer your question?
I think that's a good answer, but we'll see. I'm just looking to see if our questioner has anything else to ask for.
Can I make a quick comment, Kent? Sorry before that. So first of all, Andrew, it's a great work. We did some work on LC3 in exosome secretion from neurons, and we saw very similar things. So was really happy to see that when it's published. And my comment was that we saw that whenever there was an accumulation of autophagosomes in different neurodegenerative models. And one question that I have in the proteomic analysis that you did, it's a really cool technique. You focus on the RNA binding protein. Did you also saw proteins that are in other aspects, let's say even protein degradation, ubiquitinated ligands and enrichment of other type of pathways?
So I missed the last part. Proteins involved in degradation.
Protein degradation, ubiquitination, ubiquitin ligands, and things like that, because it could be hypothesized that this is an alternative pathway for degradation, and you should see increase in those proteins in some sense.
We saw a few ubiquitin ligases. I'm trying to remember off the top of my head. I think we might have seen one of the March ubiquitin ligases. And there were a couple others, but certainly they were not enriched. And I think one interesting thing that we think about is that, I mean, the autophagy pathway itself is actually a ubiquitin like pathway. LC3 itself looks like, at least structurally kind of like ubiquitin. And so it could be it's interesting to think that it could be functioning kind of like a ubiquitin-like molecule to sort things for secretion or degradation, depending on the context. So the fact that we saw ubiquitin itself and we saw some ubiquitin enzymes, but they weren't nearly as enriched as you might have expected from the classical autophagy pathway.
Hi, this is Anne Wayman. I wanted to ask if you thought there was any connection to any types of non-canonical autophagy like Lando or LAP, so LC3 associated endocytosis and phagocytosis.
We do. I mean, it's really interesting that 2 or 3 papers came out kind of very in a very similar time frame, kind of describing these autophagy-related pathways that are somewhat distinct from classical autophagy. And so that's actually an active area of investigation in our lab. At least on the surface, it probably looks most similar to endosomal microautophagy. From our understanding of Lando and LAP, there's not really an intraluminal budding aspect to that. You mostly see LC3 on the surface of endosomes or phagosomes, and it's directing the trafficking of those vesicles to degradative pathways. Whereas endosomal microautophagy, you tend to see an intraluminal budding event. So on the surface, it looks most similar to that, but we don't know for sure how exactly they're related. But we think probably there is a relationship.
Asma has a question.
Yes.
Asma, I think you're unmuted.
Yeah, okay. Thank you so much. I have a naive question. As you may know, in autophagy, when you induce autophagy, ATG12 is in complex with ATG5 and 16. Mm-hmm. In your knockout, do you think that this is, a non-canonical role of ATG12 alone, or, all the complex, 16 and 5 also?
So we would probably speculate that it would involve basically all of the conjugation machinery. Well, we didn't dissect all of the conjugation machinery in our paper. There's a couple other papers that have implicated, for example, ATG5 and ATG16 in exosomal secretion or production, right, or EV production or secretion as well. So our speculation would be that they probably are involved as well. And I would like to point out that, even though you mentioned ATG12, we also found the ATG7, which regulates, is kind of at the apex of this whole pathway, for the conjugation pathway, and required for all downstream events. It disrupts it as well. So it's not unique to ATG12. And in fact, I think ATG7 is a really strong a place to target the pathway because it's required for all the other steps in the conjugation.
Yes.
And if I could just interject very briefly, just related to the previous question on Lando and LAP, probably one thing really good to look at in this complex is the ATG16, because there's actually mutations in ATG16 that can actually help you discriminate between a more non-canonical pathway versus a classical pathway. And so I think that's a really good question going forward, is dissecting this whole 5, 12, 16 complex in a little bit more detail, at least from an autophagy, aficionado's point of view, yeah.
Do we have any other questions right now? I'm not seeing anything on chat, but I thought that I would ask something because, my lab has had some focus on microRNAs in the past, and Andrew, you mentioned that we always look for what we want to see. And it has been the case, I think, in the EV field for quite some time that we've focused on microRNAs, often to the exclusion of other RNAs. And I know, I think Esther would probably agree with me on that too. And I don't know, exactly what the reason for that is. There are historical reasons, and there's just the stability of the microRNAs. But when I was looking at your RNA results, I didn't see anything in the microRNA end that was jumping out at me. Did you happen to see something, or is that just you just didn't do that that particular analysis?
We did see actually microRNAs, but a lot of them actually dropped out between our two genetic knockouts. So if you actually delve into the data and you look at the ATG7 knockout, which is probably the strongest blocker of conjugation, if you will, but certainly ATG12 is also required. We find that there's a lot of microRNAs that are actually impaired in the wild type versus the ATG7 example. It didn't carry across. And of course, all these proteins are involved in things other than autophagy and have their own unique regulation. So ATG7 really potently also blocked microRNA secretion as well. So I think that there's definitely a broader role for autophagy in regulating RNA metabolism and things like that we haven't really, I think, even touched the surface of yet, given the number of RNA-binding proteins and RNAs that we're starting to see affected both inside and outside cells.
So you see, you do see some global effects on microRNAs, but I'm wondering, do you see any of the specific microRNAs that some other groups have reported to be, preferentially loaded into EVs?
Off the top of my head, I can't think of any, and we didn't, because it didn't really, I guess, was it conserved or regulated in both the ATG7 and 12 example. We didn't focus on any microRNAs specifically, but it's a good question, and I think it's something we could probably go back and look at a little bit more. I know there were a few LET7 related microRNAs that we found, and I know that there have been implicated in being secreted in various I guess post-transcriptional regulatory events, but off the top of my head, I can't recall. And we've kind of been looking more at SNO RNAs and other RNAs as opposed to micro, where a lot of other people are focused.
Yeah, I think I mean I like the experimental setup that you have here, and I think that when you look at some of the papers where people have described the specific incorporation of specific microRNAs, many times those are related to serum. And you ask the question of those labs, and they say, well, we couldn't grow our cells without serum, or without, maybe they tried to take EVs out of it, but not RNA. So we have I think we have a literature right now where there's a lot of confusion, and I think that the way that you approached it is a good way. Maybe there's more to dig out here too. So that's great.
Yeah, thanks.
Okay, we have a few more questions that have come up. So Jenna Morris Love, I believe you're unmuted now.
Hi, is my mic working? Can you hear me?
Yeah,
okay.
We can hear you.
Yeah, I just had a question. It looks like Jay actually answered it in the chat, but I was wondering if you had tested functionality of any of the RNA binding proteins or the non-coding RNAs.
Functionality in terms of transfer, or
yeah, into target cells.
We have not. We haven't actually. I think an important area would be to try and really figure out what RNAs some of these RNA binding proteins are actually interacting with, and whether they're actually transferring them, or maybe they're just being secreted and potentially being degraded in cells that kind of sort of take them up through phagosomal endosomal pathways. But no, we haven't looked at transfer to this point.
Mm-hmm. And if I could add one more question while I'm still unmuted. I was wondering if you've looked at nucleophagy at all in particular, or if you guys are kind of playing with that.
So we obviously there's a large nuclear repertoire of RNA binding proteins that are in our data set, but we also find a number of cytoplasmic RNA binding proteins like LARP1 being secreted as well. It's an interesting question, but we haven't looked at it to this point. Certainly it's on our radar, and it would be interesting. I worked on herpes viruses before, and they're known to bud from the nuclear envelope,
right,
and they can't be transported through the nuclear pore complexes, and so it would be an interesting way to kind of meld like trafficking and release. Yeah, but not to this point. It's certainly a really interesting area, I think.
Thank you.
We have a question from Tran. Tran, I believe you're unmuted now.
Can I ask, like, whether in the ATG7 knockout cells, do you see like an alternative package of non-coding RNA in the cells? Like, are they sorted to somewhere else, and if they're not released?
Sorry, I had a little bit of difficult time hearing you. I heard ATG7, and it kind of trailed off a little bit after that. Maybe you could speak a little louder.
If I can just, I can do that. In the ATG7 knockout background, do you see an alternative package of ncRNA in the cell? So you're asking if there is a cellular difference. Is that correct? Like inside the cell?
Yes. If they're not released, then would they be? If they're deficient in the release, then would they be alternatively sorted somewhere else in the cells that degraded? Or
so what's the fate of these RNAs if they're not secreted? That's the question?
Yes, indeed.
We actually we don't know, to be honest. We certainly our data is normalized in such a way that we're looking at things that are secreted and either not changing or basically secreted above intracellular changes. But we have no idea what the fate of these RNAs is within the cell to this point. It's interesting that there seems to be a large number of our RNAs and RNA binding proteins show associations with these RNA granules. So there may be some sort of connection. I think Sheckman's lab has hinted at that as well. But we don't know what they might be doing if they're not being secreted, and whether they might be targeted for degradation or accumulating granules or whatever.
All right, and I believe that Esther had a question about the vault RNA that had recently been reported as implicated in autophagy. The vault RNA 1.1. Esther, are you unmuted? Do you want me to unmute you?
Yes, I think I'm unmuted now.
Yes.
Yeah, so looking at your figures, you have only very small percentages of vault RNA, I guess. I can't really see it, but I was wondering whether you specifically looked for this small vault RNA that was published last year and that was implicated actually in autophagy.
We looked at vault RNAs and we didn't really see any dramatic changes. And so at least in our system, there didn't seem to be a role in regulating vault RNAs. I can't really speak to it very much because we didn't really find a functional role for the autophagy pathway, at least in our system in regular.
Okay. Yeah.
So, and I just wanted to follow up on Roger Alexander's question. So he had asked about what you think these EVs might be targeted to. Is there anything that you know about these EVs that might lend them to targeting to a specific cell type, let's say?
To this point, no. I mean, there's hints at things, but it would be pure speculation. We have always kind of we perceive autophagy to be a degradative pathway predominantly, and so I think there may be roles for it in transferring things to cells to be potentially degraded, but it's really kind of unclear what cells the tropism of these vesicles might be. There's definitely hints if you look through the data, but I think we're
I think at this point, one of the things, so LC3 is inside the lumen of the vesicle. And so I think in order to, kind of really do that experiment well, we need to better understand if there's something specific, at least on the exofacial surface of an LC3 positive vesicle, in order to follow it. And, actually, I know a lot of people in this, probably in this chat or in the Zoom actually use, things like nanoflow to quantify things. And I think that's where we have a bit of a limitation. But I think as we kind of get an understanding of the external surface of these, we'll know where the tropism probably, the hope is that if there's something on the surface of these vesicles that now binds to a receptor, or that, you could actually get a better sense of that. But we remain a little bit, we just don't know at this point in time, because most of what we've characterized is inside the vesicle, and as a result, it's hard to follow, where it would land.
Good. All right. Well, I think we might have a few other questions that are coming up, but we are at the hour now, and so we're going to close the journal club. Please do feel free to contact me if there's anything that you would like to present or a paper that you'd like to discuss. So I'd just like to thank Tom Driedonks for his presentation of this paper and for bearing with us when we had to reschedule. And also a special thanks to Andrew and Jay, who joined us today. And I think it's always nice to have the authors around to answer some of the questions that nobody else can answer. So thank you, everyone. I hope you all have a wonderful weekend, and I'll look forward to talking with many of you again very soon. So goodbye. Take care.
Thank you.
Thank you.